Zygote is a fertilized cell. Yeah, that's diploid.
Yeah,
okay, I'm going to start over.
Okay, well, I felt like picking that one. That's all I want to want. I want to pick
Behave.
Alright. Dogs! Alright, let me just start over with this whole process. Okay? First of all, here's a cell. This cell is a diploid cell. We use the word diploid meaning it's got two sets of chromosomes. The green set came from your dad and the white set came from your mom. If it's got two sets of chromosomes, it's called a diploid cell. What we're going to do is go through this process called meiosis, and we're going to make cells that have half that number, that only have one set of chromosomes. Those cells are not called diploid. You know what they're called?
Haploid.
Haploid, having half the number of chromosomes.
So half that right there would be haploid.
Yes, and how many are here?
46.
Well, how many are on the cell that I have on the board?
6.
6. 1, 2, 3, 4, 5, 6. So how many are we going to end up with?
3.
3 in each cell. Is that exciting to you?
How did you get 8 out of 3 chromosomes?
It's explained in the picture on the bottom of page 175.
It's too cute.
Do you have your book open?
I brought my rock.
Dang it. You can't make that. We'll do it tonight. So, we start with prophase 1. In prophase 1, the chromosomes coil up where you can see them. And then the nuclear membrane dissolves. This is our review from yesterday. And the centrosomes move to opposite sides, shoot out spindle fibers. The spindle fibers attach to the chromosomes. Jessica, are you with me?
Yes.
And then the chromosomes are going to start moving. And this is where they do something different. Do you remember what that different thing is?
They find homologous pairs.
They find their homologous pair. And these ones will pair up, and these ones will pair up, and these ones will pair up. And at that time, they're kind of laying on top of one another, actually, and exchanging information. And so that's why I have this model where I can switch the pieces. We call that crossing over. And it introduces variation to our gametes. And you have to understand that each sister chromatid will cross over many times. So this white will have a number of green lines in it. And this green will have a number of white lines in it. And same with this one, and same with that one, and that one. They all cross over. And it's kind of random what pieces switch. So keep in mind, this is a single cell that's going to make sperm cells. But in a man's testes, there are millions of cells all doing this. Guys, you make 2,300 sperm per second. And every one of them is shuffling up the genes. You're never going to end up with 2 sperm that are the same. It's like if you took a deck of cards and you threw it in the air and shuffled them all up, you'd hardly ever end up with the same order. And even more so because there's only 52 cards in a deck, or is it 50? I forget. The states and the cards. Anyway. There's way more. There's 46 chromosomes, and each chromosome can switch pieces in 100 or several hundred different ways. So you're never going to get two sperm that are the same. That's prophase 1. If I asked you this on an essay, could you write me what happens in prophase 1? Tell everything that happens in prophase 1. Bam.
2300.
Metaphase 1. What happens in metaphase?
The pairs move to the middle.
They line up in the middle. Y'all remember this from yesterday? Anaphase 1.
The chromosomes separate and move to the opposite
Chromosomes
side.
separate and move to opposite sides. Now, they can separate like this, but you have to remember that each pair can separate either way, because really they're laying on top of one another, like I told you. So this pair can go right and this pair can go left or the other way. And each pair separates independently from the other pairs. We call this independent assortment. And so, again, it's like shuffling of the cards. We're shuffling up the chromosomes during this independent assortment process, which also leads to more variation. Variation is good, because that gives our organisms a wide range of characteristics so they can survive any changing climate. That was like in the puppy video. That's as far as we got yesterday, isn't it?
Yes.
That's-
Yeah.
Metaphase.
Anaphase just happened. Metaphase is when they were lined up in the middle. Anaphase is when they split and moved to opposite
So,
sides.
prophase, they find a homologous pair.
Homologous.
Homologous pair. Then metaphase, they line up. Anaphase, they split apart.
Yes.
Telophase happens.
The homologous pairs split apart. They don't split apart at the centromere like in mitosis. The homologous pairs split apart.
Does telophase happen next?
Yes, telophase is next. Are you ready for telophase?
Yeah.
The chromosomes arrive at opposite sides. The spindle fibers disintegrate. Chromosomes arrive at opposite sides. A new nucleus forms around the chromosomes. And then the chromosomes decondense. They spread out. So you can no longer see them anymore. They go back into chromatin. And the cell starts to pinch inward. Don't you hate people who say pinch? Starts to pinch inward, and then eventually will divide in the middle, cytokinesis. And now we have two cells.
So there's. Oh, that's.
Telophase.
What did it form around already? What's this, a membrane?
This membrane formed around the chromosomes,
And then the cell has a. Okay.
and then the cell divides. It's the same as mitosis in that respect. And now we have two cells, but guess what?
We're going to do it again.
Well, count the number of chromosomes in each cell.
There's too many.
There's 3 where we started with 6. What do we call a cell that has half the number of chromosomes?
Haploid.
These are haploid cells. After meiosis 1, you end up with haploid cells. They have half the number of chromosomes.
The cells that divide again?
Yeah, they're going to divide again.
I know, but what's half of a haploid called?
It's still haploid because all we're doing in the second division is we're going to divide up these. Remember, these are exact copies of one another that we formed during interphase. We don't need 2 exact copies in the same cell. There's no reason to have two of the same thing, so we're going to split those up during this meiosis 2.
At the end of meiosis 1, do you end up with 2 cells with 40? I'm confused.
2 cells with 23 now.
2 cells with 23. Okay.
Mhm. We started with 46, now we've halved the chromosome number, and now we have 23. What do you think about all that, Savannah? Huh? Pretty neat? Okay, we good? Want to go on to the next? You want to see it in video first? Maybe that'll help. We're making sex cells here, Russell. You're sleeping through it.
I'm not sleeping. Okay.
All right, prophase. Here's prophase 1.
Prophase 1 begins with condensation of the chromosomes. Homologous chromosomes, each made up of 2 sister chromatids, come together in pairs. This pairing is called synapsis. Each chromosome pair is called a tetrad, a complex of 4 chromatids. Chromatids of homologous chromosomes cross over each other and exchange parts at chiasmata, singular chiasma. Meanwhile, other cellular components prepare for the division of the nucleus. The centrosomes move away from each other and spindle microtubules form between them. The nuclear envelope and nucleoli disperse. Finally, spindle microtubules capture the kinetochores that form on the chromosomes, and the chromosomes begin moving to the metaphase plate.
Get all that?
What's a metaphase plate?
The metaphase plate is the middle of the cell. That's where the cells will be during metaphase. Let's watch metaphase.
At metaphase 1, the chromosome tetrads are aligned on the metaphase plate. For each tetrad, kinetochore microtubules from one pole of the cell are attached to one homologous chromosome, while kinetochore microtubules from the other pole of the cell are attached to the other chromosome of the pair. Thus, the homologous chromosomes are poised to move to opposite poles of the cell.
How about that? Anaphase 1?
During anaphase 1, each pair of chromosomes is pulled apart, and the homologous chromosomes move toward opposite poles. Note that sister chromatids remain attached at their center.
Centromere. What do you mean? Are we good? Jordan, you got a question?
No.
All right, you ready for telophase 1? Whoa. Here's telophase 1.
The chromosomes finish their journey during telophase 1, and cytokinesis occurs, producing 2 haploid daughter cells. Note that each chromosome still consists of 2 sister chromatids. Meiosis isn't over yet. Remember that it consists of 2 consecutive divisions. During meiosis 2, the sister chromatids will be separated.
Are we good? All right, let's do meiosis 2. We're going to do meiosis 2. Meiosis 2 is very simple. It's just like mitosis. If you know mitosis, you already know meiosis 2. Do you know mitosis, Jonathan?
Yeah.
Well, you already know meiosis 2. You ready for meiosis 2? There's a brief phase between meiosis 1 and meiosis 2 called interkinesis, when the cell just kind of sits there for a while and it'll copy its centrosomes, make new centrosomes. Well, the centrioles are inside the centrosome.
Those centrosomes are the little proteins that attach the chromosomes.
Those are spindle fibers.
What are the
No.
things in the middle?
Centromere.
Centrioles. Down. Confuse it?
Isn't it the centromere that attaches the sister chromatids?
The centromere's right there.
Yeah, that's what you were talking
Yeah,
about.
that's what I asked.
Is that what you're talking about? That's the centromere. This is a centrosome.
And then them together is a centriole.
The centriole are two little structures that look like that are inside the centrosome. The centrosome is an area where the spindle fibers come from. And animal cells have two centrioles inside their centrosome. And plant cells don't have any.
So interkinesis is...
Remember respond to sleep? There you were.
So what is interkinesis?
Interkinesis is a brief time period between meiosis 1 and meiosis 2 where the cell is just kind of sitting there, waiting. And it copies its centrosomes during that point. You ready, Caitlin, for meiosis 2? It's been a long time. I keep saying that, and we never get started on it. Why don't we get started on it now?
Do it.
Prophase 2. These centrosomes move to opposite sides. They do it in both cells. And they shoot out spindle fibers. And the nuclear membrane dissolves. And that happens on both sides. Just like mitosis. Spindle fibers come shooting out. I'm not going to draw them all the way across the cell. But imagine that they do that. The chromosomes coil up where you can see them. The nuclear membrane dissolves, and that's prophase 2. Can you guess what happens in metaphase 2?
Grab the chromosomes.
Because of the lineup.
Well, they actually grab the chromosomes in prometaphase, but
Pull apart.
metaphase 2, they line up in the middle. Metaphase 2, line up in the middle. Metaphase means middle. Anaphase 2, Hannah, what do you think?
Metaphase or
Anaphase 2.
they split in half?
Nice.
And now their sister, or their daughter.
They're daughter chromosomes now.
And before that, they were sister chromatids.
That's right, they were sister chromatids bound together at the centromere, forming a chromosome. It's actually called a duplicated chromosome because it has two. Jessica, what happens in telophase?
Splits.
What splits? What happens with the chromosomes? Anything? Telophase? No? Okay. Russell?
Chromosomes come together and form on the opposite ends, and the cell splits in half.
The chromosomes arrive at opposite ends.
A nucleus forms a nuclear.
A new nuclear membrane forms around the
And
chromosomes.
it pinches in and separates.
New nuclear membrane forms. This is telophase and mitosis too, so you need to know this. New nuclear membrane.
And mitosis is just telophase is just the phase inside mitosis.
Yeah, and meiosis.
So all those like small things are just part of one giant thing called mitosis. Right?
You talking about mitosis or meiosis now?
I'm not sure anymore.
Copying the sex cell, making sex cells is meiosis. That's what we're talking about now.
Okay. Meiosis.
3 days ago, we talked about mitosis.
Mitosis is just...
Just a normal cell copying itself exactly.
Okay.
This is meiosis forming sex cells, which is different.
Alright. And so every time one of those cells forms, the chromosomes are matched up differently. Or details of the chromosomes are matched up differently in meiosis.
That's correct.
Okay.
Mm-hmm. The cell starts pinching inward. And now we have 4 cells where we started with 1. And the 4 cells are different from one another. Look, this one has 2 whites and a green. This one has 2 greens and a white. This one has 2 greens and a white, but a piece of a white. And this one has 2 whites and a green and a piece of a green. They're all different. That's the thing about meiosis. It makes cells that are all different from one another. That's variation. Each of these will grow a tail, and so you got sperm. Yay, we made sperm. And then I'll swim over through the woman's vagina, up to the egg. We're going to learn about it in the second semester. It's great learning about it. So this cell has 3 chromosomes when it started with 6. And this sperm cell in a human it's not 3, it's 23, will fertilize an egg cell that the same thing has happened to that has 23 chromosomes. And now together we'll get a new cell that has 46 chromosomes. And then that cell will multiply and eventually grow into a baby. And the baby will have half the chromosomes it got from its dad, and the other half it got from its mom. And there's 3 major sources of variation here. We get variation during the crossing over, when the little pieces switched. That's one source of variation. Do you remember what the second source of variation is?
Fertilization.
No, not that happens later.
Mutation.
Wait, what was the first one?
The first one, remember we crossed over the alleles? That's the first source of variation.
Independent assortment.
Independent assortment. Remember when we were lined up during metaphase 1, and then we switched them in anaphase when they moved to opposite sides? They could go either way. Do you remember that?
Yes.
That's another source of variation. Because all the greens could go one way, or all the whites could go one way, or you could get 2 greens and 1 white, or you could get 1 green and 2 whites. You can shuffle them out different ways. And that gives you a lot of variation. If you read last night, they did the math for you. In humans who have 23 pairs of chromosomes, the possible combinations of the gamete is a staggering 2 to the 23rd power. Or 8,388,608. There's 8,088,368 different combinations of chromosomes you can get just by shuffling them up during independent assortment. And then the biggest source of variation comes from the last part. What is it? Fertilization. This is important to understand. This is why there is sex. I mean, why go to all the trouble of finding a mate just to have a baby? Why not just do it yourself? Why not just bud off a little piece and have it grow into another Mr. Willis? Then I don't have to find a mate. I could just lay in bed watching TV while Mr. Willis's pop off of me. Here's the thing. If you mate with someone else from a whole different family with a whole different bunch of genes, they're going to have a whole different bunch of combinations that your family never had. And so when you combine that, that's a huge source of variation. That's why there is sex. And that will make the kids really different. Because they're a combination of you and this other person that's from a whole different family. So the kids will be really different than their parents. And thus able to survive if the conditions change. You know, if your family is really hairy and you marry someone who's relatively hairless, then you're going to have kids all across the board. And if it gets real cold, the hairy ones will live and the hairless ones will die. If it gets really hot, the hairless ones will live and the hairy ones will die. It doesn't matter what the temperature does, someone will live. And your species will survive. Isn't that great? That's what that Wimpog show was about. So, fertilization is the third form of variation. It goes crossing over, independent assortment, and fertilizations. Those are the three forms of variation. And then the basic form of variation is called mutation. That's what bacteria do.
Can we get it right?
We have mutation too. What's that?
Can that be right on the quiz?
Was it discussed in the reading?
They talked about how prokaryotic cells, the only way to variate is mutation.
I might give it to you.
Yes.
Please.
Please do.
Oh,
Please,
really?
please.
Now everybody's happy.
And then
Please.
I have to
Please.
like 10 quizzes.
All right, I'll do it.
Yay!
It's because I like y'all.
I'm so happy.
Did you read the section?
Yes.
Okay. All right, so
There was a person
I was.
with nails.
Lynn was there.
The mutation,
She had
the
great
nails
coat.
were like overgrown.
oh, really? You're reading further. You're reading further than we needed to read.
That's a good thing. Now I got
Hello.
It's in the back of the chapter.
Yeah, that's at the end of the chapter. We haven't got there yet. Okay, let's see the whole process here. Let's see the whole process of meiosis. Oh, naked people. Okay, here we go. Stages of meiosis. You ready?
Let's get this started.
Watch the whole event. Are you watching row 1?
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared, and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
That's it. Doesn't that look kind of like what I have on the board?
Yes.
Here.
So you might get a question that says something like this. How is meiosis different from mitosis? That'll make you think. You say you know, Emery?
Would you want me to answer?
Yeah. Go for it.
When they
What's the M called?
Metaphase.
Metaphase?
Metaphase, they don't split apart with just the other things. What are they called?
Okay. That's
and meiosis, the homologous pairs split apart.
That's right. You're saying when they meet,
And mitosis only has one thing.
you're saying when they meet here in metaphase and they line up in the middle, in anaphase, when they move to opposite sides, they don't split in the middle at the centromere. The homologous pairs split up.
Yeah.
That's one difference. That's good. What's another difference, Stephanie?
Wait, that happens when
In mitosis, the cells produced are identical to the cells that they came from. And in meiosis, they're different.
That's a good one. Mitosis produces 2 identical cells. Meiosis produces 4 cells, and they're all different. What's another difference? Meiosis runs through prophase, metaphase, anaphase, telophase twice. Very good, Savannah.
The cells are haploid in meiosis and diploid in mitosis.
Do it, Sarah. In mitosis, you split and you still get 2 diploid cells. They still have 2 sets of chromosomes each. But in meiosis, you end up with 4 haploid cells. They only have half the number of chromosomes. That's a big difference. There's one more that no one's saying.
The chromosomes don't pair in meiosis.
In meiosis, the chromosomes come together. The homologous pairs pair up. And crossing over occurs, where pieces are switched. In mitosis, you don't get that.
Mitosis, the chromosomes just line up. They don't pair up. They just line up.
Yeah, they don't pair up. They just line up, and then they split in the middle. And each side gets an identical piece. In meiosis, they pair up, cross over, and then split up.
So mitosis, they just come together and then cleave.
In mitosis, during metaphase, they all line up in the middle like this. They're all lined up along the middle. But in meiosis, they pair up, and the pairs come to the middle.
Okay.
And then the pairs split. That doesn't happen in mitosis. Mitosis splits like that. Yes?
In mitosis,
Yes?
can they independently vary?
No, they don't independently assort. They're all lined up in the middle, and each side is going to get identical copies. There's no assortment going on. You can only get assortment if you pair up. When you pair up, then they'll independently assort themselves. So independent assortment only happens in meiosis, not in mitosis.
I'm sorry. I'm looking at there's no definition for bivalent on here.
Same thing.
This is a bivalent. Homologous pairs, tetrad, bivalent. 3 names for the same thing. Yes, Hannah?
What makes a cell, is it just like type of cell? Is there a certain type of cell that does each one, or is it just like...
It's a cell in your gonads that does meiosis.
Are those the only cells that do it?
Those are the only cells that do meiosis, because the only reason for doing this is to make sex cells. There's no other reason for doing this, except for making sex cells. So the only place this happens is in your gonads. I just like saying the word gonads.
The sperm cells you just made, those could have been egg cells, right?
Those could be what?
Could have been egg cells, right?
Yes. This whole thing could have been egg cells instead. I wanted to do sperm cells because I'm a guy.
Is there any difference though between them?
Yes, there's a small difference I'll talk about tomorrow. Other questions? You sure? Are you guys meeting with the AP history today after school?
Nope.
I was thinking I would go over photosynthesis and respiration if you wanted to come.
Can we do that
Okay.
like a scheduled day?
Scheduled day.
Like, can you let us know ahead of time?
No. I was going to do it today.
Well, if we do show up, will you go over it?
I got it on tape if you want to watch it.
Oh, yeah.
Or we can record it today if you want, if you can't be there.
Will you?
Yes. Also, I got other good news. Our test is scheduled for Wednesday. Can't do it Wednesday. They scheduled the PSAT.
No.
And the seniors are going to help you.
Seniors are going to Halyards. They're not taking it.
Test canceled.
So I'm moving the test to Tuesday.
What? Thursday.
Can't do it Thursday. There's too many other tests Thursday.
That's a busy. I thought it was so nice.
What chapters is it?
Chapters 7, 8, 9, and 10.
Oh my goodness. It's gonna be hard.
Can I turn this off?
Actually, yeah, this was in the other.
We're going